Hello everyone. So in this lecture, I am going to start cell structure part 1 after the biomolecules. So I had started with biomolecules because you would see that even in this chapter, the knowledge of biomolecules is absolutely essential if you want to understand about the cells. So in these classes, I am going to teach you about the basic details and concepts of cell structure. However, these are equally important for two reasons. First of all, before you go over to the elaborate details of any chapter in life science, the basic concepts are absolutely essential whether you want to appear for the board, degree course, or any competitive exam. Secondly, if you go for interview to any reputed college or universities, the most questions they would ask in the interview would be from basic concepts. So, understanding the details and understanding the basic concepts are equally important. But don't worry, since these are short lectures, I will also elaborate on each and every part and I will publish those lectures also soon for the competitive exams. So for today, let's move on to the cell structure. The whole cell structure is a bit big, so I have divided it into two lectures, and today I am going to tell you about the part 1 of that lecture. So most of us already know what are cells. So let us revise once more about it. Suppose every one of us lives in a house, right? So houses are made of what? The basic unit is brick. Each and every brick joins together to make up the whole house. Similarly, for our body, the basic building units are cells. Cells are the basic building blocks of all organisms. However, organisms can be unicellular, that is made up of only one cell, or multicellular. If the organisms are multicellular, then there is a hierarchical organization. Several cells interconnect to form tissues, tissues combine to form organs, organs comprise an organ system, and several organ systems function with interconnections and form an organism. So, in science, we believe in one thing. We follow the rule that seeing is believing. Unless and until you are doing an experiment and unless and until you are seeing something, we don't believe it. So, cells are very, very small in size. Like when you look at any skin or your hair, you won't be able to see the individual cells. What you see is a structure made up of millions of cells. So with naked eye, we cannot see the individual cells. Then how do we see those? So the microscopes come into play in this respect. There are generally two types of microscopes used for studying cells. One is light microscope. So in here, the visible light passes and bends through the lens system to enable the user to see the specimen. Why it magnifies? Because it has certain glass lens which magnifies the slide objective. So two parameters are important in microscopy. These are the magnification and resolving power. Magnification is the process of enlarging an object, but resolving power is more important because how do you like to look at an image if it is very big but blurry? Even we prefer high resolution camera lenses in our mobiles, right? Similarly, in the microscopes that have high resolving power, the cells or any other images resemble very sharp and very focused images which are the crisp, clear to look at. So the resolving power is the microscope's ability to distinguish two adjacent structures as separate. The higher the resolution, better the image's clarity and detail. So this is a basic microscope, light microscope used in laboratories. These are the adjustment screws. This is the stage which can move upward, downward and sidewise. These are the objective lenses which are used to magnify your specimen. The specimen is kept here on this stage these are the eyepieces through which you look at and see your specimen. However, you must know here that cells are always transparent. So it becomes a little bit difficult to look at them through normal light microscope. So the easier solution is to color them with special stain. However, stains kill the cells, but the structures before they were killed remain intact. So you can see the colored versions of the killed cells. Next is electron microscopes. So nowadays most of the research laboratories and institutes have at least one electron microscope to look at the microscale structures or even nanoscale structures. So electron microscopes don't use light, but they use a beam of electrons. This allows for higher magnification because this whole thing operates in vacuum and also provides higher resolving power. But the method to prepare the specimen is very rigorous in case of electron microscope, and it definitely kills the specimen. There is no other way you can look at electron microscope specimens without killing them. However, nowadays there is some very advantageous electron microscopes called environmental scanning electron microscope. So they claim to look at the specimens directly in a media or a culture system in which they live, and we can make video images of the cells just moving around and interacting with each other. So that is a very interesting new thing. So in scanning electron microscope, a beam of electrons moves back and forth across a cell surface like it scans the whole surface. In transmission electron microscope, however, electron beam penetrates the cell and we can see even what is there inside the cells. So after studying the cells, let us come to the cell theory. Cell theory was proposed by Schleiden and Shivan, who were studying tissues, and proposed the unified cell theory, which states that one or more cells comprise all living things, cell is the basic unit of life, and new cells only and only arise from pre-existing cells. So these were the basic details. Now, basically, if you classify cells, there are two types of cells, prokaryotes and eukaryotes. This is a typical example of a prokaryotic cell structure. Now, both prokaryote and eukaryote share four common components: a plasma membrane, which is the outer covering, cytoplasm, a jelly-like fluid within the cell, DNA, cell genetic material, and ribosome to synthesize proteins. We would look at every one of these aspects later on. But prokaryotes also differ from eukaryotes in several ways. Prokaryotes are generally single-celled organism that lacks a nucleus or any other membrane-bound organelle. So you must realize how difficult it is for a bacteria to use its single cell for every function, like for reproduction, for division, for growth, for nutrition, for respiration. We have so elaborate and different organ systems. But all of these functions are performed in bacteria by a single cell. Amazing, isn't it? So, most prokaryotes also have a peptidoglycan cell wall, and many have a polysaccharide capsule. This polysaccharide capsule is very important for some pathogens, because what happens is this capsule protects them from antibiotics, and even if we eat antibiotic, it doesn't kill the bacteria. The cell wall also acts as an extra layer of protection, helps the cell maintain its shape and prevent. Capsule also enables the cell to attach to surfaces. For example, in our mouth, we certainly have dental times, dental cavities. Why? Because those bacteria, Streptococcus mutans, attach to our cells with the help of their capsules. That's why if we do not brush very vigorously, they don't get removed off. Flagella are used for locomotion. Pili exchange genetic material during conjugation, etc. So the diameter of prokaryotic cells are 0.1 to 5 micrometer. These are significantly smaller than eukaryotic cells, which have diameters ranging from 10 to 100. Small size is necessary for all cells, whether prokaryotic or eukaryotic. But why? See, not all cells are spherical in shape, but most tend to approximate a sphere. So from high school geometry, you must remember the surface area of a sphere is 4 pi r square. But formula for its volume is 4 pi r cube by 3. So if the radius r of a cell increases, surface area increases as a square of its radius. But volume increases as the cube. So the volume increases much more rapidly. Therefore, as a cell increases in size, its surface area to volume would decrease. The same principle would apply if the cell had a cube shape. So therefore, if the cell now grows too large, plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. So as a cell grows, it becomes less efficient. So one way to become more efficient is when the cell becomes too large, it divides. Another way is to develop organelles that perform specific tasks. So the division thing is followed by prokaryotes, the organelle thing is followed by eukaryotes. To the later evolutionary objects, that is the eukaryotic cells. You must know that prokaryotic cells came first. The eukaryotic cells came later. They are far more evolutionary advantageous. So have you ever heard the phrase form follows function? It's a philosophy that's followed in architecture. That means buildings should be constructed to support the activities that will be carried out inside them. For example, a skyscraper should include several elevator banks, have its emergency room easily accessible. Similarly, our natural world also utilizes the principle of form following function. Unlike prokaryotic cell, eukaryotic cells have a membrane-bound nucleus, numerous membrane-bound organelles, and several rod-shaped chromosomes. Because a membrane surrounds eukaryotic cells, it has a true nucleus. So that's why the name Eu means true, karyote means nucleus. And prokaryotes, pro means before, karyote means nucleus. So before nucleus and true nucleus. The word organelle means little organs. Organelles have specialized cellular function just as your body's organs have specialized functions. So you can say that a cell is also somewhat independent in itself in that it also contains small miniature versions of organs. But overall, an organism performs so many complex functions that a single cell cannot perform all those. So they interconnect with each other and forms the organism. So the plasma membrane. Like prokaryotes, eukaryote cells also have a plasma membrane, a phospholipid bilayer with embedded proteins that separate the internal contents from the surrounding environment. If you have followed my channel and if you have looked and learned with me the lipid content, the lipid chapter, you must know that this is a basic phospholipid bilayer which forms a cell. The hydrophilic tails are outside, heads are outside, the hydrophobic tails are inside, and there are proteins and cholesterol molecules interspersed between them, which makes up the total cell membrane. So the plasma membrane controls the passage of organic molecules, ions, water and oxygen into and out of the cell. Waste such as carbon dioxide and ammonia also leave the cell through this. But besides this, there are some organs in which the plasma membrane specializes in some special functions like absorption. Like in small intestine, this plasma membrane folds into finger-like projection that we call microvilli. They absorb nutrient from digested food. This is an excellent example of form following function. People with celiac disease have an immune response to gluten, which is a protein in wheat, barley and rye. The immune response damages microvilli and thus the the nutrients cannot be absorbed in these individuals. This leads to malnutrition, cramping and diarrhea. Next, the cytoplasm. Okay, before we go on to the cytoplasm, don't think that the plasma membrane is till this only. In the next two lectures, I will describe in detail about the plasma membrane model, structure and function. Since this chapter is about the cell in whole, so we are just giving some basic concepts about all the organelles. Next is the cytoplasm. This is the cell's entire region between the plasma membrane and the nuclear envelope. It is comprised of organelles suspended in gel-like cytosol, the cytoskeleton and various chemicals. So many of us think that maybe cytoplasm is a totally liquid and watery. That's not the case. It's always gel-like, so that it can restrict the drastic and abrupt movements of the organelles inside the cell. So even though cytoplasm has 70 to 80% water, it has a semi-solid consistency, which comes from the proteins within it. However, proteins are not the only organic molecules. Glucose and other simple sugar, polysaccharide, amino acids, nucleic acids, fatty acids, and derivatives of glycerol are also there. Also, there are some ions. Many metabolic reactions, including protein synthesis, take place in the cytoplasm. In the prokaryotes, however, all the metabolic reactions takes place only inside the cytoplasm. Now comes the nucleus, the basic signature of an eukaryote cell. Typically, nucleus is the most prominent organelle in a cell. Even if you look at a cell in the microscope, the nucleus would be very visible to you. The nucleus houses the cell's DNA and directs the synthesis of ribosome and proteins. So outside there is the nuclear envelope, which is a double membrane structure. Both the nuclear envelope's inner and outer membranes are phospholipid. The nuclear envelope is punctuated with pores that control the passage of iron molecules and RNA. Nucleoplasm is the semi-solid fluid inside the nucleus, like cytosol, where we find the chromatin and nucleolus. However, the nuclear envelope is very much more stricter than the cell membrane. That is because even if a toxic substance enters the cell membrane, the cell has certain bypass mechanisms through which it can make the molecule go out of the cell. However, that is not true for nucleus. If a toxic molecule enters the nucleus somehow, it may cause extensive damage to the DNA. That would result ultimately in cell death. Therefore, the nuclear envelope acts as a much more stricter sentry than the cell membrane. So, what are the chromatin and chromosomes? DNA, as we know, is the hereditary material. You must have learned in my chapter nucleic acids. Also in prokaryotes, DNA is organized into a single circular chromosomes. In eukaryotes, however, chromosomes are linear structure. Every eukaryotic species has a specific number of chromosomes, which does not correlate with the size of an organism. For example, in a human, chromosome number is 46, but in fruit flies it is 8. Chromosomes are only visible and distinguishable from one another when the cell is getting ready to divide. When the cell is in the growth and maintenance phase, proteins attach to chromosomes and they resemble an unwound jumbled bunch of threads, like this. We call this unwound protein chromosome complexes as chromatin. This chromatin condenses to form these chromosomes. This happens during the metaphase of the cell cycle. We would move over to cell cycle and I would describe to you in detail. But this is actually chromosome painting technique, which uses certain stains which colors each of the homologous chromosomes in a different color. See, here are red, purple, yellow, green, etc. So what is the use of this? Suppose a person has inherited some genetic disorders which has resulted in some abnormalities in the chromosome structure. So a cytogenetics technique can followed, which can color each of these chromosomes separately, and suppose if there is any abnormality in this chromosome, it would be easily acutely observable to the scientist. Now, ribosomes. These are the cellular structures which are responsible for protein synthesis. Ribosomes appear either as cluster or single tiny dots that float freely. They may be attached to the plasma membrane or the endoplasmic reticulum. Electron microscopy shows that in ribosomes, which are large protein and RNA complex, consist of two subunits, large and small. These two subunits are also entirely different for prokaryotes and eukaryotes, which we will learn later. So the mRNA travels to the ribosome, which translates the code provided by the sequence of the nitrogenous bases in mRNA and a specific order of amino acids in a protein is formed. I have also explained this to you in my nucleic acids chapter, but to tell you shortly, this is the ribosome, large unit, this is the small unit, this is the mRNA which has the code for the protein. Now, an amino acid is recruited each time by a tRNA which brings the amino acid and this is joined to the growing polypeptide chain. The reaction required for this is catalyzed by the ribosome. Now, one can argue that mitochondria is one of the most important organelles of the eukaryotes. So, since eukaryotic cells perform a large number of functions simultaneously, it needs a large amount of energy. This energy is provided by the mitochondria or the cell powerhouse. They are responsible for making ATP, cell's energy producing coin. ATP represents the cell's short-term stored energy. So, for example, when we eat, the glucose is converted to other intermediates and ATP is formed. In mitochondria, the process of forming ATP uses oxygen and produces carbon dioxide. So, this is the carbon dioxide that you exhale with every breath. This comes from the cellular reactions. So, muscle cells have a very high concentration of mitochondria. Why? Because muscle cells need considerable energy to keep the body moving. When cells don't get enough oxygen, they do not make much ATP. Instead, produces lactic acid, which accompanies the small amount of ATP they make in the absence of oxygen. So, have you ever noticed that when you exercise after a long period of time, your muscles cramp and they ache a time period later? That is because your muscle has accumulated this lactic acid, due to which there are pains and strains in the muscles. So that is a symptom that you have not inhaled properly, your body did not get enough oxygen, and that's why due to incomplete oxygen, lactic acid formed. So the mitochondria are oval-shaped and double-membrane organelles that have their own ribosome and DNA. In a cell, nucleus is separate, but inside an organelle mitochondria, it again has its own ribosome and DNA. So you can imagine how active it must be. Each membrane is a phospholipid bilayer embedded with proteins. The inner layer has folds called cristae. We call the surrounded area the mitochondrial matrix. The cristae and matrix have different roles in respiration, and we would look into it in the cellular respiration chapter. Now we are coming to the last part of cell structures part 1. About two small structures I would tell here, peroxisomes and vacuoles. Peroxisomes are small round organelles enclosed by simple membrane. They carry out oxidation reaction that break down fatty and amino acids. They also detoxify many poisons that may enter the body. Many of these reactions release hydrogen peroxide, which would be damaging to cells, but these reactions are confined to peroxisome and hence these are broken down into oxygen and water. For example, peroxisomes in liver cells detoxify alcohol. Glyoxysomes are specialized peroxisomes in plant. They are responsible for converting fat into sugar. Plant cells also contain many different types of peroxisomes that play a role in metabolism, pathogen defense, stress response, etc. We will look into all of this when we discuss plant metabolism in detail, but the variety of peroxisomes in plants are much more than animal cells. Vesicles and vacuoles are membrane-bound sacs that function in storage and transport. Other than the fact that vacuoles are somewhat larger than vesicles, there is a very subtle distinction between them. Vesicles are like buses. They can fuse with either the plasma membrane or other membrane system within the cell. Additionally, some agents such as enzymes within plant vacuoles break down macromolecules. The vacuole's membrane does not fuse with the membranes of other cellular components. So vesicles are smaller, they take part in mainly transport. Vacuoles are larger and they take part mainly in storage. So that was it for today, cell structure part 1. I hope you have liked the lecture. If there are any questions, you can always comment in my YouTube channel or you can comment in the Facebook page. If you have liked the video, please like it, share and subscribe. So stay tuned for the next lecture where I will describe part 2 of the cell structure. Stay safe.